Okay. Let's go to, can you see the screen? Okay. Go to the NCBI and we're going to search for a gene. Just go to the NCBI.nlm and .nih.gov. This is the longest URL name ever. And then you can type something like kinase. Suppose that I don't know anything about kinase. I just want to play around to learn more about the kinase protein. So in the NCBI, if you search for something, it's going to show you a lot of related information, like the literature, including books, uh, and the publications on PubMed, gene, and the expression data, mostly from microarray and next-gen sequencing, and the genome that contain the gene. Also, the genetics, like a SNP database, for example, and clinical association. You can also see it's like some proteins and the chemicals that related to the kinase, right? So if we care about the gene, you can just click at the gene, click at the gene here, and see what happened. So you're going to see a lot of sequences that has something to do with the kinase. For example, the first one, Pik3CA, that's the gene name. The gene name is actually named by the scientist. So most of the time, it's up to the scientist to name the gene. Right? And sometimes you're going to see that one gene has many different names. Like this one, in NCBI, the reference name will be Pik3CA, but there are some other names as well, which is the aliases, the CLAPO, CLOVE, PI3K, PI3K alpha, for example. That's why there has to be a system that can identify the gene and also the function of the gene in a systematic way, right? So that we can refer to the correct gene or to the right gene. And you can see on your right, it's going to be the results by taxon, right? So you can see these are all the organisms that has a gene. Oh, sorry. Let me go back a little bit. So these are organisms that has a gene. You can see Homo sapiens, which is human, and some other like Mus musculus is a mouse. Some other Ratus is a rat, and salmon, I guess, I don't know, and some other taxa, which is many more. So there are many organisms that need this kind of gene to function. So suppose that we just care about the human kinase, we can just click at the Homo sapiens. Okay, and you're gonna see a lot of 5,000 kinase related gene. Okay, so let's see maybe one gene that pretty much like play a role in many pathways in our system, which is the JAK2. The JAK2 is called the Janus kinase. Do you see this one, right? The JAK2, the ID 3717. It's a JNS kinase, which is happen in many pathways, including the immune system and some other systems as well. So suppose that we care about this one, just click at the JAK2, just the name of the gene. And in here you can read about the gene, you know, especially the summary of the gene. So it says that this gene product is a protein tyrosine kinase involved in a specific subset of cytokine receptor signaling pathways. That's why it's quite important in many pathways because it's about cytokine receptor. Has been found to be constitutively associated with the prolactin receptor and is required for responses to gamma interferon. So mice that do not express an active protein for this gene exhibit embryonic lethality associated with the absence of definitive erythropoiesis, blah, blah, blah. So this is, you know, just the intro of the gene. And you can see where the gene is located, right? Inside the chromosome number 9. So this is the where the gene is located. This one should be positive or negative strand. What do you think? Do you think the gene, the JAK2 is in the positive or negative strand of the genome? Anyone? What do you think? Negative or positive strand? The JAK2 gene here.
Do they on the positive strand because they are in the same way of the whole genome.
So what about other students? Do you think the same thing? You agree? So how do you know the direction of the genome?
They have the arrow on the top left and right below the chromosome 9.
Okay, yeah. Mostly we read the genome from left to right. It's just, I mean, to tell the negative and positive strand is more like a convention. It's just something that we agree upon. It's not a rule, right? Because when you have a genome, you can just say, okay, I want this part to be a positive 5 prime, this part to be a 3 prime, for example. And it's kind of more like a convention. And our convention is that we're going to read the genome or gene from left to right. So this one, the 5 prime will be on the left, and the 3 prime will be on the right, for example. Okay. I mean, according to the genome, okay, it's not the gene, because the gene we know that that's going to be a poly A tail on the 3 prime side. For the gene we know, but for the genome we don't know because there's no the 3 prime the poly A on the genome. And it depends on what sequence that we sequence from the genome. There are 2 strands on the genome, and it depends on which one we get when we sequence the genome. Okay, so that's a lot more information, and we not gonna go on over all of this stuff because I think it would be good for you to play with it yourself. And this one is the result from the RNA sequencing using next-gen sequencing data. So nowadays, this one, in the past there's no like RNA sequencing data available. Nowadays you can see the expression of this gene in different tissues. And you can see that the same gene is actually expressed in different level in different tissues. This is pretty normal. It's pretty normal because the gene play a different role in different tissue. That's why it's expressed more or less depending on its function. And this is the expression from the RNA sequencing. And you can see, because doing RNA sequencing it's quite easy to do. Right? You can just pull the mRNA, the total mRNA from the tissue, and then sequence, and then you count how many times you see the JAK2 genes, and then you get the expression level like this. Okay, let's see if we want to blast, if we want to find a homolog, the homologous sequence of this gene. You should go down to, let me see. Sometimes there will be a link to blast that you don't have to copy and paste the sequence. Let's see if we click here, blast. It's not the right one. And let's go to the left-seq protein. Okay, you have to go to the left-seq protein first. I think the protein RNA pretty much the same. I mean, it's going to take you to another web page and show you the sequence of the gene, and then you can do blast search from that. Because this one is pretty much all about like all the related information. But if you want to play with the sequence, you can go to the related information part on the right hand side, and then you go down, and then you're going to see the ref-seq gene or ref-seq RNA or ref-seq protein. And this is a suggestion for if you want to look for the homologous sequence, you should use the protein sequence as much as possible, because the nucleotide sequences will not use a metric like the substitution metrics like PAM or BLOSUM. So it's going to be different. So, and it's much better to use the protein sequence to search for homologous sequences, because as I said in the first class, that even you have a mismatched amino acid, but they could still have the same or similar property. So I still get a good score. That's why you can pick up a lot more sequences that may have some variation in nucleotide, but not the amino acid. Not much in terms of amino acid, due to the number of amino acids in the same group, for example. So it's always good to use protein sequences to do alignment or building like multiple sequence alignment for creating the phylogenetic tree, for example. So if you can, if you want to do anything with the homologous sequences, and you can find the protein sequence, use the protein sequence. So click at the ref-seq protein, and then it's gonna take you to another page. Okay, and then from this one you can do blast search, right? Find regions of similarity between these sequences and other sequences using blast. Just run blast here. And this one, you know, it's gonna insert pretty much all the sequences for you. So if you just want to search for one sequence, you can just delete the rest and keep the one that you want to search. And this one you can actually copy and paste the FASTA sequence into this text box in order to do blast search as well. You don't have to, if you don't know the accession number or the ID of the sequence, you can just copy and paste the sequence, you know, to the text box. And you can also specify, you know, what part of the gene, what part of sequence that you want to search specifically, right? By typing the position of the nucleotide or the amino acid here. So are you guys following? Okay. Let me know if you can follow, right? And then you can go down and, you know, this is going to be very important. You have to select what type of databases that you want to search for the sequence. You can use a non-redundant database. You know, this is going to be a huge database, right? Because it's going to contain pretty much everything in the world that non-redundant, the unique protein sequences in the world. And this could take you a while to actually finish. And or you can use the reference protein, which is a subset of the non-redundant protein sequences. This reference protein will be more reliable because it's curated by, you know, expertise in the field. It's already curated, so it should be more reliable. So if you just want to have a good, reliable sequences that are homologous to your sequence, you can use reference sequence protein. Or you can, you know, look at the UniProt or, you know, protein data bank or metagenomics and etc. So that means there's so many things that you can do. And this one we're gonna just choose the non-redundant protein sequences, maybe because we want to see if we can get something out of like some other animals from the search. And you know, there are so many different, you know, tools that you can use to search for the protein sequences. You can use BLASTP, a quick BLASTP, or just the default BLASTP program, or you can use SciBlast, which is going to be it's a program that is going to run many times, many rounds of searching. So you get you can get more sequences basically. It's going to increase the sensitivity of the search, but you're going to get a lot more sequences that might not be specific. So this one is going to build some sort of the position specific model to search, and some other, you know, tools that if you work on the protein a lot, you know, you should be familiar with all those stuff. But I don't usually. You can ask Atanapat or Japrasit about, you know, this kind of tool because they may have used them to do the protein search for the structure analysis. Okay, and I just want you to see a little bit, you know, at the algorithm parameters. This is a part that most users will not go to. They will not even know that, you know, you can actually click and play with this part, because it's more like an algorithm parameters, and you can control how the algorithm works by just changing the numbers. And the first one is quite important because it's going to limit the output. Right? If you want to see more output, you can select like 500, for example. and it's going to show you a lot more output, like a lot more matches. But if you select only a 100, it's going to show you only the top 100 sequences. Because sometimes you just like, why I only see a few sequences, like 100 sequences, because you select like only 100. Right? But if you want to see a lot more, which is includes some low scoring sequences or alignment, you can just choose like 500, for example. So it depends, you know, but mostly 100 would be a good value to start with. And this is a very important, like I said before, that the word size is the one that BLAST used to look for the sequences in the database. And if you have a larger number of the word size, it means that the sequence is going to be more specific. But if you use the tool, it's going to be less specific, so it's going to take more longer time. But it could be, you know, you should get, it should be more sensitive, but it's going to take a lot more time. You can try that. But 6 is going to be kind of more specific, so it's going to take less time to search. And you can play with, you know, just a single sequence. You can try to, you know, vary the parameters, and you see what happens. And the expect threshold is actually is more like an e-value that I'm going to explain later, but you can set this number as well to select to set a cutoff of the output of the output of the alignment. And again, this one you've seen this one before, right? The matrices that you can use, and just choose, you know, if you want to just look at the closely related animal, can change, for example. And the cost, you know, this one I would not recommend you to change at all unless you know what you're doing, because this is about the gap, right? It's gonna give minus 11 every time that it finds a gap, and every time you extend the gap, you're gonna get minus 1, for example. So the first gap will be 11, and the second consecutive gap, you get the minus 1 every time. So this is, you know, you can play with this one, but it's gonna change the result of your analysis by a lot. And this one is, let me see, I don't quite remember. So if you don't know what it is, what the parameter is, you can just, you know, click at the help and then see. Like this one is the amino acid substitution matrices may be adjusted in various ways to compensate for amino acid composition of the sequences being compared. The simplest adjustment is to scale out substitution scores by an analytically determined constant. So this is more about, you know, how you compensate for the composition of the amino acids in your sequences. Like the composition means like how many A's, how many G, how many Y, how many T are in your sequences. And that could actually affect the scoring metric, right? So that's something that I think you just leave it like this, because if you don't know what it is, you can leave it. And the last thing, the filter, I think nowadays you don't even need to do, you may not need to check all these checkbox anymore, because BLAST actually, BLAST is doing a good job now to kind of, you know, ignore all the low complexity regions, for example. So in the sequences, for example, the gene or the genome sequence, you're going to find a lot of regions that has a very low complexity. Like it could be like A, G, 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 A, for example. So it's just, you know, it has no variation in that region. It's just a repeat of A, repeat of G. So that's the low complexity region, and that's could, it could give you a high score when you do the alignment, because, you know, A match with a lot of A's will match with A, for example, but it doesn't mean anything. So BLAST is kind of, you know, ignore or filter those regions in the alignment to give you a much better result. But, you know, I believe that nowadays, you know, BLAST will do that pretty well, even though you don't check or don't tell it to do. Okay, let's go on. I just click BLAST and see what happens. So this is gonna take a while, and you can wait because we are searching for the non-redundant protein database, and this is gonna take a while, and it will limit um, the size of the database. You know, it's gonna run faster. Also, if you limit, if you change the matrix, you're gonna get you're gonna get the result faster as well. And this protein is kind of long. You know, I forgot to look at the length. It's like 900 or 800 amino acids, so it's quite long. So it's gonna take a while. Are you guys done? Hello. Are you guys done? Not yet? Okay. We can talk, you know. I don't hear anyone. It's like I'm talking by myself. Okay, you can look at my results in the meantime to wait until your result come up. This is my result, and you know what? It changed a lot, you know, like from last time I look at the result page. Nowadays, you can do, you know, you can filter, you can do so many things that you couldn't do before. So this is quite new to me as well. I mean, the look of the page is new. And basically, you can see this is not surprising, right? Because um, what should match 100%, you know, with your sequence is your own sequence, right? We use the JAK2 protein sequence from humans, so we get exactly the same thing back, and that's going to be 100% identity, right? So that is not surprising, and you still get you also get the accession number that you can go look at. And you can see, you know, there's another JAK kinase that you can click and look at. This is quite, it's just a little variant of that sequence. It maybe, you know, it's only like a single nucleotide mismatch. This could be, I don't know, maybe a polymorphism in human, because, you know, I don't know. I mean, there are two of them in the database that actually match. The first one matched 100%, that's our sequence. And the second one matched like 99%. It's only a single amino acid that actually different. Anyway, but when you look at the positive, the number here, the positive means that amino acid is also still the same. It's similar to the amino acid that it's substituted. So it means that, you know, the change doesn't do anything much because it still get the same type of amino acid. Okay, so and these are the tabs that you can click, right, to see the description. This is all just the name of the sequence that matched. and also the score, total score, and those kind of things. And then you can see the graphic summary of the match, right? So if you see the red color, it means that the query sequence is our sequence actually matches those sequences very well, more than the score is very high, higher than 200, basically. You can see the legend up here, say the alignment score lower than 40 will be black, 40 to 50 will be blue, blah, 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 and more than 200 will be red. And a bonus that you get, an extra bonus you get from doing blast nowadays, that you get the domain function for free. These are all the domain function on the gene that you get for free. And you see, there are two domain that there's a PTK Jack2 and PTKC Jack2, RPT1 and RPT2 domains. It's a PKC-like superfamily, right? And you can, click at the protein domain and see what it is. So the information is all connected, right? So you can see here about the PKC, PTK-like protein family and what it does, right? So this is a lot of information. I think it's just too much information, but it's a good thing, if you want to learn. And then you can see the alignment, if you care to look at the alignment. But this one, is quite good that, I think the top matches will give you a very good alignment, maybe from end to end, because they're all the genes and they can align, or the protein sequence, and they align very well. So it's, perfect. Most of them are perfect. You don't see any different from your query. And for the taxonomy is also pretty useful, because you can tell that, oh, my sequence is from human, for example. And you can see it matches sequences, 14 sequences from human pretty well. The score is like 2000 something. But if you look like down the taxonomy tree, you're going to see that the score in some organism is not that high. And let me show you maybe some animals that I know. Look at the bison. Well, the bison is a cow, right? It's even told ungulate, which is a cow. So you can see there are two sequences from bison that actually matches our sequence, but it's not very, very well. And actually these tools are all predicted. They're not verified from the experiment, but they're just predicted amino acid sequences. So the score is not very high. So maybe our JAK2 in human and bison could be somewhat different. So this is very useful. There's a lot of useful information that you can get for free from doing the blast search. See, you can do so many things. Okay. And I want to explain a little bit, if I remember correctly, the total score is basically the score of all the sequence alignment. And the max score is basically the max score from the best score from the region that has the highest score. Because when you do the local alignment, you can have more than one region that actually align to the sequence. Some of them, if you have only one region, like this one, a single region, the max score is going to be equal to the total score. But if, sometimes you have maybe two regions, you don't match like the entire sequence, but just part of the sequence that match. And maybe there are more than one part, and you're going to just calculate the score by the max score by picking the best score, the one that has the highest score. So the max score and total score might not be the same. And then the e-value, this is more like a statistical number that show you how likely it is that the match actually happened by chance. So how likely it is that your sequence will match random sequences in the database. So this is showing you that the 0 means that the probability is so low, like very, very low. So 0 doesn't mean that it couldn't happen, right? It's not like 0 possibility. It's not like that. It's just that the probability is very low. So it's the probability that your sequence match this sequence by chance is very low. Because if you, for example, you search, you have a short sequence like 100 amino acids, and you search in a very huge database, a very, very big database. Chances are that you're gonna match something just by chance. They are not homolog, but just by chance happen to be similar. Right? Some sequences just happen to be similar, but they are not derived from the same common ancestor or whatever. So this is a good number that show you, okay, this should be the real deal. This should be a real homologous sequence, because it's not going to happen by chance, but the E value is very low. 0 is like very low. And this is what we say the expected threshold. If you set the expected threshold to 10, it's going to ignore like any sequences that has like the E value higher than 10, for example. But these all sequences are valid, should be fine, because the E value is very low. It's very low. Okay, just for fun, I'm gonna give you one sequence to try to blast and see what you get. Maybe I'll give you this as an assignment, because I don't think we're gonna run out of time. I want to do another thing, which is the primer design, before we end today. And I think you can do the experiment by yourself for just an assignment. Okay. So I think the blast. Any question about this one? It's pretty straightforward. Once you understand the concept, right? You understand how the sequences are aligned, how they are scored. You know about the matrix. It's pretty straightforward to understand the result. And now you should be, I said before that now you should be aware of all the pitfall, but you can also interpret the result. Any problem? Any questions before we move on to the next topic? And I'm going to ask you to do this as an assignment, and I think you're going to get a much better picture of the concept that we discussed. Okay, so the next thing, I would like you to go back to the NCBI again. NCBI. No, just NCBI. No. So this time, instead of searching for sequence in the database, we're going to use another tool, to design the primer for the qPCR, for example. And this one is going to utilize the sequence alignment search tool as well. So basically, it uses BLAST under the hood. We don't have to do that by ourselves, but it's going to use BLAST to do the job. And they actually has some other like features that can help us design a good primer for our experiment. So this is something that I think it's going to be useful, because most of your advisors will expect that you can do primer design after taking the bioinformatics course. Actually, it's pretty simple, and as I said before, the tricky part of designing a primer is going to be the biology of your gene or your sequences, not the tool. Right? If you know better about the biology of your research of your organism or gene, you can design a much better primer. Okay, go ahead and maybe what gene do you know that you would like to try? Anyone of you has a good gene name in mind that I can try? Just one gene, tell me.
Cas9.
What? Cas9?
PARP9.
Okay, I don't know what it is, but let's see. Let's see what it is. PARP9, PARP9. Okay. It's a poly ADP ribose polymerase family member 9. Wow. Okay, I haven't heard this gene before. Well, let's go to the gene again, right? And this time again, we're gonna go to the reference sequence. But this one, we're gonna go to not the protein anymore because we want to design a primer for the RNA sequence. Because if you want to do the qPCR, for example, you would like to amplify the RNA sequence. So we're gonna go to the RNA-seq transcripts, the rep-seq transcript. So when you do the PCR, when you design the PCR, you should stick with the reference sequence. Don't use some other sequence, some other like submitted sequences, because the rep-seq is more reliable. As I said before many times, that it's kind of verified by experts in the field. So you should use the rep-seq as much as possible. This is kind of long. The sequence is quite long. It's 3,400 mRNA, and you can, there are so many different variants, the transcript variants. So we're gonna use just one. You can try different variants, but I'm gonna choose the variant 1 for myself. Okay. You have to go to this page, right, to see all the information. And on the right hand side, if you go down, you're going to see the product of the mRNA, right? This is, and also the sequence of the mRNA. This is a GenBank format, right? This file format is a GenBank format, and you can download and do something with it later. But for now, we're going to go to the primer design tool, right, by clicking at the pick primers. Just, the one that below the run blast comment, the link, you go to the pick primers and you click the pick primers. See, it's pretty much like blast, it's gonna insert the ID, the accession number of the gene into the text box for you, the input box for you. So you don't have to do it yourself. And then it's going to ask you to, where you want the forward primer to start and to end, where you want the reverse primer to start and end. And if you don't have anything specific in mind, you can just skip that. And also, if you have your own primers, for example, you already have your own forward primers, you can just insert the forward primer in here. Again, if you have a reverse primer you want to use, you can insert the reverse primer in here. But if you don't have anything, just leave it blank. And then the PCR product size. This one is quite big because the minimum is 70 base pair, but the maximum is 1,000. That's too big because mostly for the qPCR, it shouldn't be more than like 100 or 200. Also, this one you can just adjust according to your need, right? So it depends on what do you want to do. And this is going to give you maybe 10 sets of primers. You can increase the number if you want more. And for people who work on PCR, who work with PCR, can you tell me what should be a minimum melting point, melting temperature for the PCR process. Do you think these numbers are okay, or too low or too high? Any idea? Hello, any idea? Do you think
I think it should be 55 to 65.
so? What's the reason?
I think it can be lower than this, and it can be higher than this a bit, because it's quite far from the expansion temperatures.
Okay, so I don't really know much about this one, but I've heard that you can, for the qPCR, you can, increase this number a little bit. It could be something like 60, and optimal could be like 62, for example. But as I said before, this all depends on what kind of things that you want to do. You have to check with someone, about the setting. Appropriate setting, and this is the max TM difference, means the melting temperature between the forward and reverse primers. What's going to be the difference between these two? Because two primers could have different melting temperature depending on the sequence, and this is going to limit that difference to be only 3 Celsius. And you can also choose to have the program to design, to try to get the primer that actually span across an exon-exon junction, or you can limit the primer just to be within an exon, not across exon junction. So if you have maybe 2 exons, and your primer could span across that exon. So this is all depend on what you want. But if you don't have any preference in mind, you can just choose no preference, right? And let the program do its job. And this is going to be a little bit more detailed, like how you want your primer to match the exon junction. And this also the intron length range, like, okay, I don't quite remember. You can click here. This specifies the range of the total intron length on the corresponding genomic DNA that would separate the forward and reverse primer. Right? So this is the one that you want. For example, if your primer is going to be across the intron, like one primer is on the other side of the intron and the other primer on the other side of the intron, you're going to have to, you can specify like how big is the intron that you want to be in the middle. So this is one is up to you. You can change, or you can just leave it like that and see what you get. And the important thing is that you can select the database for the primer to search against. Like you can test your primer against the reference sequence mRNA to make sure that your primers will not match anything else except the gene that you are interested in. Or you can look at the NR, which is a non-redundant database. That's going to be a much bigger database. So in that case, you're going to check whether your primers will match anything in the world, for example. So this is it's up to you, and you can choose what you think is suit you best. And also you can select the organism, right? So if you only work with human, you can just limit the specificity searching for human alone. Like you don't care about animals. You're not gonna use this primer to amplify any animal genes. You can choose to limit your specificity testing to the human only. So the program will check for you that your primers will amplify only the human gene, not other animals. But if you work with other animals, you can add, you can add more organism, to the list if you want. I believe that you can type. Yeah, okay. You can type the name. It's going to show, and you can choose, like, if you want, your primer to check against the house mouse, and it's going to show you like what genes that will be amplified by your primers in mouse, for example. Okay, let's leave it like that, just the human. We don't care about anything else. And this one is the max target size, like how you want your the maximum target, the size of your target. So it's going to be the amplicon size for PCR target to be detected. Okay, let's try. There's also some other advanced parameters, but I don't think you should. Some of them are actually from BLAST. You see, it actually uses quite, bigger word size than before, because now the primer is actually kind of short. It's only 20 nucleotide or amino nucleotide long. So when you have a short primer, you want to increase the word size so that you're gonna get kind of more specific sequence than before. Because I said before that when you search for the short sequence, you're gonna have a lot of chance that you're gonna get a non-specific sequence. That's why this one they use a bigger word size to increase specificity. So there's so many other things that I don't have any idea what they are, because it's just about the details depending on the genome and gene that you work on. So if you work on something that's different from human, like some bacteria that grow in somewhere deep under the sea, for example, you may need to check all these parameters to make sure that your primers will have you have a good primer. Otherwise, you can just go by with the default settings and see what you get. And this is going to take a while. Just have to wait. Are you guys done? Mine is finished, but waiting for uploading data. Okay. Okay, so here is what I get. Now, this is the first time I've seen this gene, so I believe I can go right and left. No. I can't move to the left, but Okay, so this is why I get the primer size is 20 and here's all the melting point is like 62, which is the optimal because we set it as optimal at 62. And the GC content is like 60, 65, which is, I think it's okay. And the self-complementarity is the chances that these two primers will actually bind to themselves, you know. So if the primers can bind to themselves, that's gonna be a problem because it's gonna, you know, instead of binding to the template, it's gonna bind to themselves, and you cannot. It's not going to amplify any of your sequence. So this is good, you know. If the lower the number is the best. So you want this to be very low, like 3 or 6. And you can see from down here that the products on intended target, which means that your primers will actually bind to these sequences like the Homo sapiens, variants 1, variant 3, X2, X1, blah, blah, blah. So this, all the stuff, because, you know, we design the primers from the variants 1, but we didn't specify that we want the primers to actually to exclude all the variants. So right now, my first set of primers will actually match like all the variants of the PAF9 gene, right? And this is the set number 2. So now you have like a 10 set of primers that you can hopefully choose which one that you want, and then you can go ahead and synthesize and use it in your experiment. So are you guys okay with that? You get the result, the same result. So let me go back and check a little bit if I can exclude some variants from. Okay, actually down here you can see that's a choice, that's an option that you can choose to allow splice variants. So this one, you know, the program will try to design primers that will not bind to other variants, so other like splicing variants from the gene. So you can get the exact same isoform or the variant that you want to amplify. So let's see if I choose this one and if I can exclude all the other variants that I don't want. Have to wait a minute. Okay, I think the result. Okay, this one actually is still, my primers actually pick up the variant 1, but still, it also still pick up variant 3. I think this is because the program cannot do better than this, so it cannot exclude the variant 3. Maybe it's kind of difficult, I think. Because sometimes if the isoform is very similar, it's going to be very hard for program to get only a single variant. So this one, I believe the first variant and the third variant is actually highly identical, it's highly similar. So, you know, none of my primers can actually exclude the variant 3. So this is something that you're going to run into, right, when you do the experiment. When you're going to get, if you want to just measure the expression of the variant 1 using the qPCR, you're not going to get that, because this is, anyway, you're going to get the variant 3 as well. So you're going to count both variant 1 and variant 3. Okay, so this is something that, it's something you can see right away when you use the bioinformatic tools. So so the person that told me to use the PROP9, could you tell me who is that? Could you tell me like, have you ever done like a PCR or something to this gene before?
Yeah, I used to perform PCR on this gene.
Okay, so what were you looking at? Like, were you looking at the expression level, or were you looking at looking to clone the gene?
To clone.
So how did you design the primer that time?
I don't know, because my advisor gave me that primer.
So any of these primers look like, do any of these primers look like what you have?
What?
I mean, do any of these primers that we get from BLAST, primer BLAST, look like what you already have? Okay. Yeah, you could check that. You could look at the result here and compare with the one that you already have, and see, see if there is something that similar. There might be some, I don't know, because we don't know how you get the primer. So I believe that if you want to clone, it depends on, I don't know, if you want to clone the gene, you may actually have to amplify not only the gene part, right? I think you have to amplify more than, I mean, the outside of the gene as well, the DNA outside of the gene. It's not going to be mRNA, it's going to be the gene, right? The whole gene, including intron and exon. And also some like upstream sequence and downstream sequence. Am I right? How did you clone the gene? Did your sequence like include upstream and downstream sequence?
I clone all the gene sequence.
The DNA, right? The DNA sequence, not the RNA, not the mRNA. Right? Because if you do cloning, you have to clone the DNA, not, it's going to be a cDNA, not the RNA, right? And the cDNA actually include exon and intron, and also the untranslated region, I think. Otherwise, you cannot express the gene inside the bacteria. That's my guess anyway. So that's going to be different. What I want to say is that if you want to clone the DNA, you have to go with the reference gene, with the cDNA, and then you're not going to clone at the gene sequence like this. This one is for like measuring like to count how many times you see the mRNA in the sample using the DNA amplification, the RNA amplification. But if you want to do cloning, it's going to be different, because the cloning, you have to think about what should also be inserted into the plasmid or the cell, the genome of the cell, which could include something else, not just the gene, right? It could include some other leading sequence like untranslated region, upstream, downstream, those kind of things. And that's going to be a lot. I mean, the application is different, so the way you design the primer is going to be different. You need the genome sequence. You need to know like where is the untranslated region begins and ends, so that you can pick the entire region to decide the primer. Actually, I can show. Let me see if I can go back. Not sure about. So how many of you are doing the cloning right now? How many of you are doing cloning, the gene cloning? I'm just curious. I think it's quite difficult, you know, the gene cloning. Sometimes you just don't know why they don't work, why it doesn't work. Let me see, part 9. Okay, if you go back to the page that we search for the gene, we can see the Genome Browser. So this one is pretty useful because if you want to see where the gene is located on the genome and how and what the structure of the gene look like, you know, you can go here and look at the Genome Browser. Okay, this is quite complicated. Let me zoom out. Okay, so this is the gene, right? The gene sequence, and it actually includes exon and intron. So if you want to do design the primer for cloning, I believe that you have to, you know, select the entire gene and also some sequence upstream of the gene and some sequence downstream of the gene as well. Because, you know, the upstream of the sequence and the downstream of the sequence, most of the time is going to contain some untranslated region that actually important for expressing the gene, right? It actually regulates how gene is expressed in the cell. So you have to, you know, select not only the gene sequence, but more than that. Okay, so I'm actually not familiar with the NCBI Genome Browser that much, but usually when I want to get some sequence, I used to go to the Ucsc Genome Browser. So this is another genome browser that you can use, and you know I use this one a lot because I can upload my own sequences to visualize on this genome browser. It's a Ucsc genome browser, genome.ucsc.edu. And in this one, I'm just going to show you how I can extract the gene sequence, and then I can do something else like the PCR or design a primer or something. And you can search, for example, PARP9 from here and go. I already chose the human assembly, so this one should give me the PARP9. You see, I can select the NCBI RepSeq curated subset, so I just choose the one that more likely to be the real one. Okay, this one has variant 1 as well, so I just go with this one. So this one looks pretty much like the one that you saw earlier, but you can see the difference here a little bit, you know, different. I can move to the left. I'm going to show you this one. you can drag and select the part that you want. So this is pretty easy to learn how to use it. It's quite user friendly, and you can see the thick line here represent the exon, and the thin back line and blue line here represent the untranslated region. So the protein coding sequence ends at this point, because this is the last exon, right? Because this one is on the reverse strand, so this is the last exon, and this end is the 3' end, and you can see there's a long, a little. It's not that long; it's quite short, but there's a part of the gene that is not actually translated into protein, but it's called the untranslated region. On the other hand, if you look at some isoform, you're going to see that some isoform has a very long. This one is like very, very long untranslated region at the 3' end, and the last exon actually not at the same location, right? This one has the last exon here. On the other hand, the last exon of these isoforms or splice variants ends here. So this is actually what happened, when you have when you study gene in human, many times you're going to have splicing variants, and they're all expressed in different tissues, for example. So sometimes you just have to know like which one that you want. And you can actually, this one I just zoom out, and I can just select the gene and go to the sequence. You see, if I select the gene, I can, see all the information, including the expression level of the genes in different tissues, and looking at the 3D modeling of the protein and blah, blah, blah. Right? There's so many things that I can see. Pretty much the same on the NCBI database. Right? You can, I don't quite remember. There's actually a link that you can get the sequence right away. Sometimes, the website changed the interface a little bit, so it's quite difficult for us to find what we used to find. Okay, so now I click at the reference sequence, and it takes me to another page that has some sort of information about the sequence. And I can look at, the predicted protein if I want to download the protein sequence, or I can just get the mRNA sequence, or I can get the genomic sequence, which is the gene sequence, the DNA, which is the gene sequence. So if I want to design a primer for cloning that maybe like may cover the whole gene, for example, I may want to get the genome sequence here, and then I can tell the primer blast to like put the primers, one on the other on the end of the sequence, and the other one on the top, the beginning of the sequence, to include the entire gene. And in the Ucsc Genome Browser, I can select, like if I also want the DNA, which is the an untranslated region of the 5' end, and also of the 3' end, also the CDS, which is the coding sequence, or also the introns. And I can also expand my DNA sequence like 1,000 bases upstream of the DNA of a gene, and also downstream another 1,000 bases of the gene. So this one, I'm going to get 1,000 bases before my gene, and then a gene that includes pretty much everything, like 5' untranslated region, CDS, 3' untranslated region, and intron. and also another 1,000 bases downstream. And then, you know, I can design a PCR to actually amplify the gene from the genome, for example, that include gene and something else around the gene, the flank, the gene. So this is something that, you know, you can do, okay, then you can copy and paste, you know, these sequences to BLAST to design the primer, right? And you can tell BLAST, like, you know, where do you want your first and reverse primer to be on the gene, on the sequence. Okay, so this is just one way that you can extract the sequence from a DNA for you to do something with it, right? To align, to search, whatever. And actually I'm going to try to show you something. Suppose that I have a sequence which is the PARP9, but just part of the PARP9. I can also go to the tool and I can do BLAST, which is which is, you know, it's like BLAST, but it's not really 100% the same. But it's another tool that you can do sequence alignment on the website, the Genome Ucsc Genome website. And this one, you know, you can search for the sequence and then visualize on the genome right away. So it's pretty useful when you have a unknown sequence and you want to know where it aligned to and how. So this one you see that I get a lot of matches, and the first one that I get is in chromosome 3, you know, starting from this position on the chromosome 3 to this position, and on the reverse strand, negative strand. And I can just go to the browser and see how it looks like. So this is my, the black, like the thick black line here is my sequence, and then I can zoom out to see where they align to. Okay, so you can see, you know, my sequence, because it's the DNA sequence, right? Because I just got the DNA sequence, and I asked Ucsc Genome Browser to extend my sequence down upstream, 1,000 bases upstream. That's why I get, you know, 1,000 bases upstream, and then also the Intron. So this is pretty accurate, right? I can map back to the same gene, the PARP9, but my sequence include untranslated region, upstream, and also the Intron of the sequence. So if you have like an unknown sequence, you know, you can come to the Ucsc Genome Browser and use BLAST to align, and you're going to see right away like where your sequence match and how. So this is pretty useful tool, and I use this tool a lot, you know, when I did my PhD thesis. They also have, you know, so many things on, I mean, on this Genome Browser, you can see many tracks. So, and each track actually represents something that really useful, right? For example, you can see gene on other animals. So you can see that, you know, the sequence actually conserve or not in other animals, for example, by looking at the line. Yeah, so many useful information, and you can actually hide or show some tracks so that, you know, it depends on what you want, and it's getting, you know, it's increasing all the time. I mean, every time the number of features that you can be visualized on the Genome Browser. And this is the first, this is one thing that we talked about maybe a day ago, the other day, you know, it's the EST tag. And I just want to show you how the EST tag look like Let me show full. So this is a human EST tag. I told you that the EST tag is the mRNA that people sequence before by they use a technique that can put a poly A tail, put a primer on the poly A tail of the mRNA and then the sequence that mRNA. So you pretty much gonna get a very good sequence on the one end of the mRNA. Okay, here's how the EST tag look like. You can see it's not long, but you get the exon, right? You get all the exon because to sequence the mRNA. So you can get some exon, and then you can kind of align them to see how the gene look like. You may not get the entire gene, but you can kind of tell, okay, how many exons are there, and how the gene starts, how the gene ends, for example. See, it looks pretty much like gene, but it's just a fragment of the mRNA that they sequence from the express sequence tag. Okay, any question about today? The tool is pretty simple, I think, but the understanding of how to use the tool properly is quite difficult to grasp, I think. But the tool is pretty simple. But I think it's going to be better for us to be able to use the tool and interpret the result in the right way, and you don't get it doesn't mislead you, to the wrong direction if you don't know anything. It's going to be an assignment, and I'm going to give it to you maybe by tomorrow evening, because you can take a rest today, and then tomorrow you have another class, which is the D network, and then you can spend like weekend plus Monday to do the assignment. And my assignment would be very easy, and just one assignment, simple assignment, and then we're gonna come back and meet again 1 p.m. on Tuesday, right? Tuesday is holiday, but I think we can still have class. just 3 hours, it's a long holiday, but unfortunately it was announced later. But if you have any problem, you can talk to Adam Basong and see if he can rearrange the class. But I think it's not going to be, I think it's going to be kind of difficult for him too, because it's quite hard for me to organize, classes for every teacher that has his or her own schedule. Okay, so is there any question? Is there anything you want to do more? Okay.
I have a question about the recording video. Where can I to access this?
I'm going to upload it to the YouTube, and then I'm going to send you a link. Actually, it should be posted on Google Classroom, if you can get to Google Classroom. Maybe I'm going to upload it tonight or tomorrow. The last 2 days, the lecture from the last 2 days. Yeah, the first day, there's no record for the first day.
Because I think this assignment I need the e-learning before do it.
Okay, but it's not going to be. I mean, the tool is pretty simple, but it's going to be more about how you interpret the result. Yeah, using the knowledge in biology. But you can talk. It's not like you can discuss and do the assignment together. I don't mind that. Okay. I mean, as long as you learn, I don't really. I have to say that learning is your responsibility. So as long as you learn, you can do whatever. Like you can talk, you can discuss, you can watch YouTube, you can do so many things. Just make sure that you learn something, because the learning is actually your job. Is your responsibility. Okay. I mean, I graduated from the US, so I'm kind of familiar with the US, the American style. In America, students are so, I mean, they work a lot harder than this, and they're kind of, they have to be more responsible because most of the time, the professors will not be available after class or outside classroom, and most of the time it's going to be like teaching assistant that help the students. And unlike in Thailand, you know, in the US, the students actually contact, most of the time they'll contact professors and they'll contact the teaching assistant to help them to ask questions or whatever. So they are more active, you know, in terms of doing assignment and make sure that they understand something. They really, really want to understand something. It's not like in Thailand, you know. So I think because you are in graduate study level, you should make sure that you understand. Don't worry that don't you know, we all get paid to make sure that you learn something. So don't worry that you're gonna disturb anyone. Just make sure that you can make us like make an appointment with me or with anyone to make sure that you know the material. Yeah. And you have to be more independent. I mean, more responsible for your own learning, because once you graduate, you know, nobody's gonna like teach you anymore. So pretty much all the stuff that I learned after I graduated my PhD is just from myself. You know, I learned pretty much everything by myself, and I don't learn about all this stuff in my PhD. I actually learned it by myself, a lot of stuff. So yeah, I think most of the time, you know, from now on, you're gonna have to learn by yourself. So make sure that, you know, you read, you study, you watch, you listen as much as you can. Because, you know, there's so many things that I don't think we can cover, like all the stuff in class. You know, the class is just pretty much like a map that you know where you should go, but it's not gonna tell you everything, right? We cannot discuss everything because we don't have a limited time. Because, you know, we can discuss a lot more about so many things, but because we have limited time, that's why we have to limit the amount of information that we can discuss in class. But so I think mostly it's going to be, I mean, what learning takes place is going to be outside classroom. So even though we are finished, you know, even though we are finished with the sequence alignment today, it doesn't mean that, you know, we don't talk, we can't discuss anymore. Okay, we can come back and you know, after G network, you know, on Wednesday, we're going to come back to the sequence alignment again, and that's going to be a new topic, which is the multiple sequence alignment. We're going to use different tools and we're going to build the phylogenetics. So the basic knowledge that we learn in this class is going to be used again on Wednesday. The reason that I don't put two topics, you know, next to each other, because, you know, you're going to be so like it's going to be so boring for you, because, you know, you learn all the sequence alignment for 5 days in a row. That's going to be too much, I think. So you're going to have like a 2-day break to learn something that quite interesting. The G network is quite, I think it's much more interesting than the sequence alignment, I mean, for me, you know. Okay, we are done today, and I'm gonna post the video as fast as I can, and then we'll talk again on Wednesday. Okay, and I'm gonna send you the assignment maybe by tomorrow in the evening. Okay, I'll see you later.
ขอบคุณค่ะอาจารย์
Thank you.